morning, everyone. My name is Suprita Verma, and I am from Delhi University, a third-year student from Biochemistry Honors. And my topic for presentation is proteomic analysis of serum extracellular vesicles from the biliary tract of infection patients to identify novel biomarkers. Now let's talk about the authors. There are several authors involved in writing this article, and the few of them are as follows. She's Jung Chang. His background is medical research and clinical practice. Yung Ning Huang is an expertise in digestive diseases. Yang Bo Liu specialized in hepatobiliary and pancreatic disorders, whereas Yi Zhang is involved in emergency medicine specialist. Peng Hua, who is also emergency medical and clinical research, and Xian Shan Huang, his background is clinical emergency care. Wei Yang in digestive disease research and treatment, and Tung Ying Chiang is focuses on digestive health. And now let's talk about their major contributions in this particular field. First is proteomic analysis expertise. Now, it basically includes the utilization of advanced proteomic techniques to study extracellular vesicles. Second, biomarker discovery. It basically leads to the identification of novel biomarkers for biliary tract infection. Third, which includes clinical relevance, which means that it contributes to early diagnosis and improves patients' outcomes for biliary tract infection. Fourth, interdependency collaboration. It includes integration of expertise from digestive diseases, emergency medicines, hepatobiliary surgery, and dermatology and sleep medicine. Now, fifth is research and clinical synergy. It includes combining clinical practices and medical research to enhance the diagnostic and therapeutic strategies. Next, we will move to the background of the literature. We will see its background. Now, this article explores the use of proteomic analysis on serum-derived extracellular vesicles to identify novel biomarkers for biliary tract infection, that is BTI, which is a most significant abdominal disease, which is associated with high morbidity and mortality rate. Morbidity means having a disease or symptoms of a disease. It is a state of being unhealthy, whereas mortality rates, as we all know, it is the number of death in a population. Now let's discuss some key points in summarizing its context and the significance within the broader field of the study. And these are as follows. First is clinical relevance. BTI, that is the biliary tract infection, it encompasses conditions like acute cholecystitis and cholangitis. It is basically characterized by inflammation and infection of the biliary tract. Now, we have seen that the current challenges necessitates the discovery of specific biomarkers to enhance early detection and management. Second is the role of extracellular vesicles. These are small vesicles which are released by cell containing proteins like proteins or lipids or nucleic acids, which reflects the cellular conditions, and hence it contributes to intercellular communication. Now we will see that they offer a promising avenue for biomarker discovery due to their role in disease pathogenesis and stability in bodily fluids. Third is proteomic approach. It employs label-free quantitative proteomics, known for its unbiased and high throughput nature, to analyze EVs from BTI patients and healthy controls. This method also enables the identification of several proteins expression profiles that may serve as diagnostic biomarkers. Now, fourth, we will see identifying biomarkers. The few identified biomarkers found in this research is CEA, CAM1, and CRB3 through Gene Ontology, GEO, and Kyoto Encyclopedia of Genes and Genomes pathway analysis. Now, fifth, clinical implications. This findings propose the CEA, CAM1, and CRB3. These are biomarkers as candidates for further clinical validation as BTI biomarkers. Their potential utilizes lies in improving diagnostic accuracy and therapeutic strategies, ultimately enhancing the patient's outcome. Now we will move into the next one, that is the introduction part. This includes biliary tract infection, that is BTI. It is a condition of acute cholecystitis and acute cholangitis. These are the key conditions within the biliary tract infection, and it is caused basically by the infection and obstruction of small interlobular bile ducts. These infections lead to inflammation of gallbladder walls also and bile ducts, resulting in diagnosis like acute and chronic cholangitis. Now, second is extracellular vesicle. Let's move on this. These are, as we discussed earlier, these are bilayer delimitated particles. These are released by cells carrying cargoes like lipids, nucleic acid, proteins for extracellular exchange. Now, interest in EVS is due to their roles in intercellular communication, immune responses, and cellular homeostasis and pathological progressions too. Let's see proteomic profiling techniques. These techniques enable high output, unbiased, comprehensive proteins, annotations from biological samples like cells, tissues, or serum. Now, serum-derived EBS. This peripheral blood proteomic analysis face challenges due to complex serum composition, including albumin and immunoglobulin. Therefore, serum-derived EBS have emerged as a preferred source for developing proteomic-based biomarkers identification. Now we will discuss about methods and material required. The first step in this is serum collection. Serum collection is first taken from participants, that is 23 PTI patients and 13 healthy donors from Zaiman Chang Gung Hospital were taken in an interval of August 2020 to August 2022. Then their further validation with 10 PTI patients were done and 10 healthy donors were taken. Now, average age was taken, that is 49.1, that is between age approximately between 27 to 87 years. Here also distinction is made, that is 19 males and 17 females. Then second is serum sample collection. Samples were not collected anywhere. It is collected in emergency department. Why? To minimize antibiotic interference. Then via centrifugation at 2500 G force for 50 minutes, a platelet-free plasma was obtained. Second, now ethics. This was important to make sure that participants' consents were taken before taking their samples. Now, second is the EV extraction via ultracentrifugation. Blood sample was diluted with phosphate buffer saline, then again centrifuged at 2000 G force for 30 minutes at 4 degrees Celsius. Then supernatant that was collected was recentrifuged at 10,000 G force. That is, G force is the unit of centrifugation. Most familiar unit we are is RPM, that is revolution per minute. Here, second one is G force. Then supernatant was centrifuged and for 45 minutes at the 4 degrees Celsius. Then filtration through 0.545 micrometer syringe filters. The ultracentrifugation at 100,000 G force for 1 hour was done. Then EV pellets were resuspended in phosphate buffer saline and stored at minus 80 degrees Celsius. Then we will be moving to next one. That is the third one, that is sample preparation. It was done basically by two pathways, that is lysis and digestion, and the purification was done for them. In lysis and digestion, we will see that EV is lysed with 1% rapid in ammonium bicarbonate. Then it was boiled with dithiotretol, alkylated with iodoacetamide, and centrifuged. Then trypsin digestion overnight was done at 37 degrees Celsius. Now we will move for purification process. It was filtered using 10 kilodalton spin filters, washed with urea, and ammonium bicarbonate. Then it was desalination desalinized using stage tips and drying using a speed vacuum. Then fourth step is transmission electron microscopy, that is TEM. Negative staining TEM was done here. EV is diluted in uranyl acetate deposited onto copper grids and stained with uranyl acetate. Its image were captured using TEM, that is transmission electron microscopy. Then fifth one is the fifth process or procedure we follows is nanoflow cytometry measurement. Its analysis was done. EV is strains. EV means extracellular vesicles strained with FITC labeled CD9 or CD81 washed and then centrifuged, and again it was recentrifuged. Then EV is concentration, size, surface protein were assessed using NFCM analyzer. A most common NFCM analyzer is N3OE nanoanalyzer. Now we will move into the next one, that is LCMS or MSSA, that is liquid chromatography and mass spectrometry. Here peptide separates using RP, HPLC column and eluted with acetonitrile gradients. Data were acquired with orbit trap fusion lumos using MS full scan and MS2 scan. Seventh step is data analysis. MS data, raw data analyzed with proteome discoverer aligned with Swiss human proteome database were analyzed. Then GEO and KEGG analysis for protein functions and pathway enrichment was done. Next is the that is 8.1 is Western blot and ELISA. Using Western blot, EBV proteins were extracted, denatured, separated by SDS phage and transferred to PVDF membrane. Primary antibodies like CEA, CAM1, CRB3, CD9, CD63 and CD81, CTSG101. These are the primary antibodies. Secondary antibodies, anti-rabbit and anti-rat HRP conjugated protein bands were quantified using ImageJ software. These were found on the software. Then using ELISA, that is enzyme-linked immunosorbent assay technique, CEA, CAM1 and CRB3 protein levels were determined using ELISA kit. The ninth step that we follow is statistical analysis. Here, students t-test and with p-value less than 0.05 were considered significant value. Then 10th is ethical approvals, that is institutional review board approved by Diamond Chang Wong Hospital, adhering the declaration of health safety. Informed consent was obtained from all participants. Without their consent, nothing is possible. Now, next we will move into the results. What we see here. Let's see. Now, extraction and characterization of EVs was done. Sample collection again, that is the same procedure is following. Platelet-free plasma from healthy donors, HC N is 3 and BTI patients N is 3. That is number of patients. And then EV isolation and characterization was done. STEM confirmed the cup-shaped vesicles in both SC and BTI samples. And NFCM showed EV diameters. HC becomes 79.23 plus minus 1.63 nanometers, and in BTI patients it was shown that 75.31 plus minus 5.28 nanometer was observed. EV concentration was found in HC patient 4.74 into 10 to the power 9 plus minus 3.82 into 10 to the power 9 pico per ml, and BTI 15.4 into 10 to the 9 plus minus 6.11 into 10 to the 9 pico per ml was observed. P is the value here, P is a constant, P is 0.0625. Then EV surface protein CDT9 and CDT81 confirmed by NFCM. Next, we will see proteomic analysis of EVs. LC, liquid chromatography, mass spectrometry, or MS data. Here we 1,659 common patients in BTI samples were identified, and 1,597 were identified in both SC and BTI patients. 62 proteins are uniquely present in BTI samples, and 192 differentially expressed proteins, 46 upregulated, 146 downregulated were observed. Top 15 up and down regulated proteins documented in ExoCarta databases were also done, and heat map visualization clusters of different proteins was done. Next one is, we will see, here is the data. The first one shows the sample C shows the heat map, whereas in figure B, it shows the red one, that is the points that are shown in red is the shows the upregulated in BTI or EV sample, whereas the blue shows the downregulated in BTI or EV sample. Next, we will moving to the next one, that is GEO and KEGG analysis. GEO, that is gene ontology, and KEGG, that is Kyoto Encyclopedia of Genes and Genomes. Pathway analyzer. These are the pathway analyzers. Now, function annotation: 67% proteins involved in cell localization were observed. 8% were involved in single organism process, and 9% in positive regulation of biological processes. Predominant roles in protein binding: 78%, and binding activities 12% were observed. 66% associated with membrane-bounded vesicles, and 18% in membrane-bound organelles, and 7% in extracellular regions were observed. Now, protein-protein interaction in protein-protein interactions. Enriched PPI network identified, correlated with KEGG pathways, citrate cycle, fatty acid elongation, complement and coagulation cascade, fatty acid degradation, and fatty acid metabolism were observed. Next, we will moving to the graph or pie charts we obtained from the graph. This shows the figure A. That is, in figure A, let's see. BP represents the biological processes. Here the in BP, 3% shows, that is the red one. That is the least. It shows the regulation of biological quantity. And 67%, that is the brown one, it shows the localization, its localization among the cells, and 9% shows the single organism processes. In second figure, the majority, that is 70%, shows the protein binding, 12% shows only a single binding, and 6% is unknown. In third figure, C, 66% is membrane bounded vesicles, 7% shows the extracellular region part, and 18% shows the membrane bound organelle, whereas 2% shows the membrane with 2.242 E minus 0.4. Now we will moving to the next slide, that is validation of biomarker candidates using Western blot analysis and ELISA validation. In Western blot analysis, an increased expression of CEA-CAM1 and CRB3 in BTI patients validated by Western blot were observed. Then Western blot density of CEA-CAM1 and CRB3 were quantified using showing higher levels in BTI patients. Via ELISA validation, it confirms the increased expression of a CEA, CAM1 and CRB3 in serum of BTI patients, consistent with the Western blot results. Now, here we will see the results that have shown via the statistical analysis. The first one shows the CEA, CAM and CRB3 were obtained via Western blots. In figure 3, it shows the densitometric quantification of CEA-CAM1, whereas the figure C shows the densitometric quantification of CRB3 biomarker. And figure D shows the CRB3 that was detected using ELISA analysis, and figure E shows the CEA-CAM1 biomarker using ELISA analysis. Now we will move to on conclusion part. This Now we will conclude that this studies highlights the utility of label-free quantitative proteomics in identifying potential protein candidates within extracellular vesicles derived from serum of BTI patients. It represents an inauguration exploration of EV biomarkers for BTI diagnosis using unbiased proteomic analysis. The finding unveiled a spectrum of differentially expressed proteins with the CEA, CAM1, and CRB3 emerging as a promising candidate for clinical diagnosis. Evaluating and exploring the molecular mechanism of these proteins promise a deeper understanding of BTI initiations and progression. These insights have the potential to signify the advanced therapeutic approaches for BTI patients, ultimately improving the clinical outcomes in BTI management. Here are the references that you have taken. Now, thank you. Thank you very much.